Okay, so goal for today, or tonight, I should say, is again for those of you that weren't here when I explained to go over just what's going to be on module one of the assessment. So not everything we did this entire year, just what the main focuses of tomorrow are going to be. So I'm going to share my screen, maybe. Let me make sure it's pulled up. Oh, someone else is joining. Module one. All right. Now let me try sharing. There it is. Okay. So you should be able to see this, and this is something some of you collected from me. I've had it on my Schoology. Um, so something that might look familiar to you all. I think it's just an easy way. Sorry. Poppy. Think it's just an easy way to make sure I cover as much as possible. So here is listed the four anchors that they will consider the main focuses of tomorrow. So first one is basic biological principles. So that was the very beginning of the school year, just saying what the difference between a prokaryote and a eukaryote is, what even is biology, and then interpreting relationships between the levels of organization. So what's an organelle versus an organ, or what's a population versus a community. So we'll go through those. Chemical basis for life is the chemistry unit. So that was water properties, macromolecules, reactions, and enzymes. Bioenergetics is just photosynthesis and cellular respiration, and talking a lot about ATP. And then homeostasis and transport is talking about the cell, parts of the cell, and then the cell membrane and how things get in and out of the cell membrane. And just as I'm going through this, again, I don't want it to be super, super long, but if you have questions about anything in particular, there's something you want me to go over a little bit more, you can either throw something in the chat for me to see and respond to, or you can unmute yourself as well, whichever works best for you. Okay, so I'm going to start with the basic biological principles. Oh, someone else is entering. Sorry. Here we go. Okay, so you probably will not have to full on label a diagram if I had to guess. Now, I'm not going to 100% guarantee that because they should surprise me every year. But usually they don't focus as much on that. So prokaryote is on my screen here. And then just quickly, eukaryote, okay? So prokaryotes, remember, are your bacteria. And the main thing you need to know about prokaryotes, and I've emphasized this a lot in my class, is that pro rhymes with no. There is no nucleus. So this red squiggly things are, oh, I thought it had a pointer. Okay, these red squiggly things are the DNA. Okay. The outside of a prokaryote usually is pill-shaped. Another way students often remember the shape is based on the fact that a pill starts with a P, prokaryote also starts with a P. Okay. And they are just very, very simple in nature. So they don't have any membrane-bound organelles. The only organelle they do have is the very simple one, the ribosome, which is necessary to make all the proteins that the DNA encodes for. Still have a membrane. Sometimes they have a cell wall, sometimes they don't. And they usually have these structures that help them move around, like the flagella, which is the tail, and the cilia. As I said, eukaryotes are more complex. So we have plants and animals as our main two that we focused on this year. So U rhymes with do. They do have a nucleus. Again, that's the main thing you'll want to distinguish between the two groups. And obviously, they are much, much more complex. So you can see a list of all of the organelles here. I'll do a very quick rundown of those. So cytoplasm is just the gel on the inside. Mitochondria, powerhouse of the cell. Okay, we'll talk about that one again when we get to cellular respiration. Ribosome, that's where proteins are synthesized, as I already mentioned with prokaryotes. The ER, I would just know smooth and rough ER are the two different classifications. The difference is that the rough ER has ribosomes, the smooth ER does not. Okay, so the rough ER is usually more associated with proteins. The smooth ER is usually more associated with lipids and carbohydrates. Where was I? Nucleus, obviously in a eukaryote, that's where all the DNA is stored. I'm gonna have to kick my dog out or find a toy for her. Plasma membrane, that's the membrane that surrounds it. Lysosome, that contains enzymes that we're going to again talk about, that breaks down waste within the cell or recycles it. Come say hi to the students. You don't need to bark at the guinea pigs. There we go. And then the Golgi is the one that is going to take things that were made in the cell, like proteins, and it's going to package them up into these things called vesicles, which are like the little packages, and it's going to send them to where they need to go. So oftentimes that's out of the cell, but it could also be within the cell itself. Same thing for most of these with plants. The main differences you will see with plants is that they have chloroplast, which makes sense because that is the organelle responsible for photosynthesis. They also are going to have this large central vacuole, which is going to store mostly water, but also some nutrients as well. This is very important, not only just to keep the plant hydrated during days where it's raining versus not, but it also provides support to the cell by expanding the cell wall where it can be supported instead of the plant where it might start to sag there. Keep in mind, animal cells do have a vacuole, but they don't have large ones like this. The animal cell vacuoles are very, very small. Okay. Let's see. Next, I'm actually going to pull up a picture. Pull up a picture, and I'm going to put my dog outside the door real quick. Come here. Probably gonna bark now because she's not in here. Okay. So levels of organization. Here she comes. It lets me pull it up. If you ever cannot hear me, let me know. This is really the first main Zoom I'm doing at my new house. And the Wi-Fi can get sketchy here sometimes. Biological. There it loaded. Okay, so I'm gonna pull up an image. I just want to make sure everyone is familiar with these. Not a good image. All the pictures are so tiny. Okay, that works. Okay, I'm just gonna share and hope it starts looking clearer. All right, so hopefully you can see kind of my little Google search there, just the fastest way to get an image. So biological level of organization. Yeah, that one's blurry. We'll use this one. So with this, you should just be somewhat familiar with the order in which they go. So all the way at the bottom that's not on here would be atoms and molecules. Okay, so atoms, the smallest, molecules being multiple atoms put together. After that, you get cells. Smaller than cells is the parts we just talked about. So the organelles. Think organelle means little organ. Okay, so organelles are inside cells. Cells make up tissues. Tissues make up organs, so the organs that are within ourselves, other organisms. All of our organs make up organ systems. So we have like our digestive system, we have our respiratory system to help us breathe. So anything that has organs all used for a similar function would make an organ system. Organism, I think I've seen a lot of students mess this up this year. An organism just means one animal, one plant. So I am one organism. My dog, Poppy, that's trying to still play with my guinea pigs and got back in the room. She is an organism. The guinea pigs are an organism. Okay. Population is next. That also is one of the most mixed up is with community. Population is one species, so a population of humans versus the population of guinea pigs. Okay. And then a community is all of those together. So my house community has humans, guinea pigs, a dog, soon to be Pepper again, the bearded dragon, and so on. Ecosystems, all the living and non-living things in an area. So everything I just said, and then also like the rocks, the sun, the nutrients in the soil, water, all of that stuff. Biome comes in between here, which is an area classified by the climate, so how much rain it gets, what the yearly temperatures are there seasons, are there not, what plants are there, what animals are there, and so on. And then biosphere, think bio is living, sphere is circle, okay? So the biosphere is just the Earth. It is talking about the Earth. All right. So I'm going to flip back now to the PowerPoint. There we go. So that was biological organization. I also want to go through now the chemical basis for life. So chemistry. So first thing with that, start this unit. The most important element for biology is probably carbon. Carbon is in all of these four organic molecules, and it is very important because it makes four bonds, which might not seem like much, but those four bonds are what allow carbon to be that main building block for all of life and support all of life. Making four bonds, as you see here, allows it to connect to things in many different shapes. So carbohydrates are usually in a hexagon or ring structure. Lipids are often in a straight line or branch structure. Proteins kind of take the square shape or line structure as well. And then nucleic acids take a little bit of everything in there. So diverse structures allow for diverse functions. Before I get into each of these four in a little bit of detail, there were two types of reactions that you should be familiar with. There's dehydration synthesis and hydrolysis or hydrolysis, if you want to break those words apart. So dehydration synthesis is exactly what it sounds like. When you dehydrate something, you are, if you think about yourself, you're probably thirsty because you are removing water or there is not water. So taking out water, and then synthesis means to make. So you're taking out water to build a molecule. So take out to build. Whereas hydrolysis, hydro means water again, but this time you're adding water. And lysis means to break. Okay, so think an L, lysis. Okay, you are splitting that bond to create now again two separate molecules. Okay, so dehydration synthesis is to build, hydrolysis is to break. All right, next. There's a lot going on this chart. I'm going to summarize the most important things. So this unit was the first one where I think everyone was like, oh my, bio can be hard, which it can be. There's a lot of information, but if you break it down, it doesn't need to be that bad for this part of the unit. So carbohydrates are primarily sugars. If we're thinking about food groups, it's like breads, pasta, again, sugars, so from fruits and anything like that. Each of our macromolecules were broken down into monomers and polymers. Monomers being one building block, so think one Lego brick if you're building a structure there, or one piece of the puzzle. Polymers are the full structure, so the finished puzzle or the finished Lego house or whatever it is that you're building, or in this case, the finished carbohydrate, lipid, protein, or nucleic acid. The monomer for carbohydrates is monosaccharide, which means one sugar. Glucose was our big monosaccharide. We talked about it in photosynthesis and cellular respiration, and it's just the main sugar of life and supporting life. Our polymers could be multiple things. There was cellulose, which is a structural carbohydrate in plants, starch, chitin, which is found in like the backs of the bugs that have like the little shell on their back, so like the rhino beetle and things like that. As I said on the previous slide, these are usually long chains of like hexagon looking shapes if you are asked to identify it. And these are long chains of just each hexagon's one monosaccharide. The function of carbohydrates is short-term energy. So short-term doesn't mean like you use it within 5 minutes, but some of them and most of them usually get broken down within a 24-hour time period entirely, which is considered really fast. All right, so that's carbohydrates. Lipids. Lipids are our fats, which can be solid or liquid. Liquid would be things like vegetable oils, primarily, whereas our fats would be things you'd find in animal meat, waxes, and all of those things. The monomer, there is really no true monomer for lipids, but sometimes they throw in fatty acids like they did here, because oftentimes lipids are going to have multiple fatty acids and glycerol as a backbone. So usually these are just repeating chains of carbon and hydrogens, as you can see here, very tiny. Okay, so just carbon and hydrogens all the way down the chain, and then usually there's a glycerol here. Okay, the function of lipids or fats, if you're thinking of it that way, is long-term energy storage, so that when you run out of energy from things like carbohydrates, you can still make ATP and have energy to pull from. Proteins, the monomers are amino acids. You should be pretty familiar with this one, given that we did a unit where we talked about how amino acids are linked together to make proteins. Okay, and the polymer is just the polypeptide or proteins themselves. The biggest function, okay, is going to be with the enzymes that we talk about quickly, but also they can be used for structural components. They can be used to do pretty much anything, as you can see listed here. And then finally, nucleic acids. You should be pretty familiar with the two polymers here. We have DNA and RNA. Your monomers are a nucleotide. So nucleotide, you can see down here, is made up of a phosphate, a sugar, and a base. Our bases, which that's not the main part of this module, are still listed here. So you have A, T, C, and G for DNA, and A, U, C, G for RNA. I'm going to get more into the base pairing and everything like that for module 2, because that is the assessment that is going to be on. Okay, and obviously that's used for genetics. Okay, enzymes. Every year I've been teaching, Enzymes have been such a big part of at least a few questions. So please make sure you are somewhat familiar with these. Enzymes are, again, a protein, and they act as a catalyst. That word catalyst means that it is going to work to speed up a reaction. So the way that it speeds up a reaction is it's going to lower your activation energy, which activation energy is exactly what it sounds like. Activation energy is the amount of energy needed to activate or start a reaction. So over here, they have the little table. The solid line is without an enzyme. So over the time period, it's taking a while to get started. The upslope is the started part, whereas then it then gets going once it is started. Whereas with the enzyme, it takes a lot less energy and therefore a lot less time for the reaction to get going. So enzymes are definitely important. Without enzymes, we wouldn't be able to digest foods properly. We would have difficulties with certain foods, which is a big reason people have issues breaking down certain types of food like dairy, like different types of carbohydrates. Okay, so it usually comes down to enzymes. So enzymes can build or they can break, and enzymes do not get used up. So once this reaction here is happening, it doesn't just, the enzyme doesn't just go away. It can be used over and over and over again. Now, enzymes only do work in specific conditions, which you can see down here. So if it gets too hot or too cold, or it gets too acidic or too basic or too neutral, whatever the pH level might be that it works best in, it is going to denature, which you can see here is when it just kind of starts to crumble apart. It's not going to work anymore. Okay. The spot where the substrate fits in, so the molecule that's going into the enzyme is called a substrate. That is called the active site. That's where the activity takes place. Okay. Moving kind of fast. Again, I just don't want to take up the entire evening. But again, if you have any questions as I'm going through this, please don't be afraid to ask. All right, that's chemistry. So we have 2 more units. From my experience, believe you just need to know the basics of photosynthesis and cellular respiration. So photosynthesis is the first process, and we usually start with photosynthesis because that is where all life on Earth originates or gets its energy from. All life, as it says down here, depends on the fact that photosynthesis can be done to create food. Okay, so think photosynthesis, think food. We are creating food here. And that food is in the form of glucose. So I mentioned in the previous 2 slides that glucose is our most important carbohydrate for biology, and this is why. All food is originating from this. You'd think she'd be used to them by now, but she's not. So when you're looking at the chemical equation for photosynthesis, I often tell students to think about if you're trying to take care of a plant, and I understand not everyone has a green thumb, but what would they need? Okay. And the one thing that they definitely need, and something that can be very bad if you forget to do, is they need water. So water goes into the photosynthesis equation. And also, plants don't breathe oxygen. Okay. They create oxygen for us. So they instead are breathing in what we breathe out, which is carbon dioxide. So water and carbon dioxide are going into it, as well as sunlight, okay, which is necessary. And that's going to create oxygen, as I just said, that we then breathe, and glucose, that food that the plants can then use or other animals can use from eating the plants. Okay. She's eating guinea pig food. Was that, no, not too many slides. Okay. Cellular respiration we are now going to take that food, break it down, and use it to create ATP energy. So ATP is our cellular energy that allows us to do so many different things, different reactions, um, and anything our bodies really need. Keep in mind, one thing that's often forgotten is ATP and cellular respiration are not used just by animals, but plants do cellular respiration as well. So it's not just animals, it's also plants. So don't forget that because imagine you're cooking food, okay? You cook the food, but then you just don't do anything with it. That's not doing anything for you. So it's the same for plants. They're creating this food in the form of glucose, but if they don't break it down, they don't eat it, then it's not doing them any good. So it kind of follows that pattern. Okay. Um, Let me respond to this. So with cellular respiration, there are two main types. There's the type that occurs outside of the mitochondria, and then there's this type that occurs inside the mitochondria. Just know that outside the mitochondria, it's called anaerobic or no oxygen. Okay, and it's going to create a very small amount of ATP, so not a lot of energy. This is also called fermentation, so something that our bodies do when we are running low on oxygen, like when we're exercising, or that things like bacteria do as well. Bacteria only do fermentation. Aer Aerobic, sorry, respiration is when think air is involved, so oxygen, and this is going to produce large amounts of ATP. So we want this done, and again, this is in the mitochondria. When you look at the two reactions, they're considered inverses of each other. So what went into one comes out of the other. Okay, and the only difference is sunlight is used in photosynthesis going in, and ATP is coming out in cellular respiration. One reminder about ATP is ATP stores its energy in the bonds. So ATP stands for adenosine triphosphate, which the key word there is triphosphate. Tri means three. Phosphates are just groups of phosphates. Okay. So you have three phosphates. When you break off a phosphate, that is when energy is released. So when we're creating ATP, we're adding the phosphates together. And when we're using energy, we're breaking it apart again. So it's a cycle of building, breaking, building, breaking. All right, we've reached the last section. So last section of this assessment is the homeostasis and then therefore also cell membrane section. Homeostasis, just think of it being a regulatory process that all types of organisms use to maintain an internal environment within themselves. So for us, some of the main ones, I always throw out the body temperature one. However, other ones can be oxygen levels or hydration levels. So if your body gets too hot, as the example is showing here, sensors like on things like your skin are going to alert the brain, which is going to cause the section of your brain that deals with body temperature to trigger a signal throughout your body to try to cool down, which that signal is sweat, usually, for being hot. Okay, and your body's going to keep going through that cycle until it cools down. Oxygen, think the same thing. You could be doing this when you get out of oxygen. You don't just breathe the same. You start to breathe faster and faster and faster. Okay, and the purpose of that is so that you are getting more oxygen within your lungs and throughout your body. Water, same thing. If you get thirsty, okay, you might have to go downstairs to your kitchen, like I did right before this, and grab a water bottle, which is going to, again, you're going to keep feeling thirsty, keep having a dry mouth until your body is satisfied. Okay. Okay, so I did get a question, and I'm just going to answer this to the group. Um, it's was asking how they're going to ask questions about homeostasis. Um, that's hard because I don't know what's on the test this year. Usually they will ask something about like which of these describes what homeostasis is. So like basically asking for which one of these is the definition for homeostasis. Or it might ask something like which of these is an example of homeostasis within an organism. So just think if they do that, if it's the definition, it's what is the set point, okay? Or if you're looking at examples, what example shows an organism trying to maintain something within itself? So if it's like water levels, temperature levels, oxygen levels, carbon dioxide levels, plants do things to keep carbon dioxide at the correct level. So things like that. Usually they aren't the most difficult questions. They're pretty straightforward with them from all the ones I've seen. So I wouldn't worry too much about that as long as you know the basics about what it is. Okay. And so finally, one way our cells maintain homeostasis is through its cell membrane. So I'm going to talk a little bit about the parts of the cell membrane first, and then I'll get into all the different ways things are transported in and out of the cell. So parts of the membrane, big part here, whoa, zoomed in really far, okay, are our phospholipids. We love phospholipids. They're cute, okay? So they have the phospholipid head and the phospholipid tails, which are going to create a bilayer, meaning two layers, okay? So we have, let me see if I can write. That says highlight. Maybe. Oh, there we go. Okay, so I can draw now. So we have, oh gosh, I'm writing on a keyboard. So we have our bi layer. This is when I wish I was doing this on an iPad. Okay, you can understand what that says. Okay, so we have our bilayer, so two layers of phospholipids. And then we have the part that loves water, which are the hydrophilic heads. Okay, so those are going to point outwards because there's going to be water in and out of the cell. Now, the inside part, the tails, I'm going to try to draw a broken heart, sad face, okay? The inside does not love water. It is hydrophobic. It is afraid of water. Okay, and therefore it is going to move away from where there is water, which again is in and out of the cell. So it is going to point inwards. That's creating kind of like that dead space where there's no fluid passing through it, separating the cell from the external environment. Also in the membrane, you will often see proteins, which are here in the green and purple. Those can be protein channels, like the green one, where things can just pass in and out like a tunnel, or they can be ATP activated proteins, where you can see this is not open on the outside of the cell here. Okay, so for things to pass through, it's almost like a toll booth, okay, that you would be driving through on the highway until it is given the signal to open up. You are not able to pass through, or molecules are not able to pass through. Okay. So that leads me to the different types of transport that there are. So our main two groups are passive transport and active transport. Passive transport does not require energy. That's underlined right here. Okay, and we have three main types. There's diffusion, facilitated diffusion, and osmosis. Diffusion is just small, very small polar molecules, or nonpolar molecules, sorry, that are going to pass directly through the membrane. So that's shown in this first image where molecules are able to actually pass through the cell membrane itself. Okay? That is often small things, again, like carbon dioxide, oxygen, much, much, much smaller molecules. Facilitated diffusion means that it is still going through without energy, Okay, but it is doing so through a protein. So it is going through a protein channel. Okay, both of these are from high to low concentration. So it's going with the concentration gradient, which is why no energy is needed. If you are pushing a ball down a hill, okay, no energy is needed to roll that ball down. Once it's rolling, it's just going to keep going. Okay, same with osmosis. Osmosis is only for water. So do not say osmosis is for any other thing but water. Okay, so osmosis is water across the membrane. It can be through primarily proteins, specifically like this green one, where they are called aquaporins. Okay, and the thing with water is that it is one of the most important transfers in and out of the cell because it is going to maintain an equilibrium between solutes like sugar and salt, as well from the external environment to the internal environment. So this is where we got those fun words hypertonic, hypotonic, and isotonic. I don't know where the eraser is. Oh, there it is. Okay. So hypertonic is when there is higher salt, sugar, whatever it might be, outside of the cell. So it's like if you drink salt water, this is what would happen to your cells. Okay? It's going to dehydrate them. It's going to remove water from the cell because it is trying to balance with the external environment. It's trying to make the external environment less salty by water leaving. Okay, so hypertonic, water leaves the cell. Hypotonic is the opposite. This would be like you drinking tons and tons and tons and tons of pure water. So they do this in hospitals sometimes if they're given an IV. Okay, they have to monitor how much they're giving you in the IV. Otherwise, your body's going to become too pure with water and not have enough solute. So we can avoid things like this happening where the cell is going to burst because water is going to start flowing into the cell rapidly. Isotonic is good for us as animals, okay? We like having an equilibrium. So we like having equal flow of water in and out because the cell and the external environment are balanced, okay? So plants actually like hypotonic, even though animal cells do not. And neither of them like hypertonic because it's just going to dehydrate both of them, shrivel them up. All right. So that's passive transport, those three. And then active transport is, I don't like that there's not really a name for it. It's just called active transport as well. But active transport through proteins, which are specifically related to protein pumps. Okay, so this is the one I was talking about with the toll booth. So this purple one, when ATP is added, okay, it's going to open up to let the molecule pass through. Okay. These are going to pump things against the concentration gradient. Okay, so we have our hill here again. Instead of it going down the hill this time, we're going to be pushing a ball up the hill, which requires energy. Okay, so we're going against the gradient. Other types that do this are endocytosis and exocytosis. These involve the cell membrane, in that the cell membrane is actually going to engulf the molecule, or it's going to fuse with a vesicle or a package, okay? And it's going to release it into the external environment. So endocytosis, think entering, En for enter, okay? It is going to literally eat the molecule, it looks like. It kind of looks like it's going to swallow it there, okay? So the cell membrane pinches inwards to form like a little cave where it can surround it. And then a vesicle is formed from the membrane through that. Whereas exocytosis, again, the Golgi will package up whatever it is that's being sent out. And those vesicles are going to fuse with the membrane, as I said, and release it. Think EX, exocytosis, for EX, exit. All right. Took me a little bit longer, but that is my really quick Module 1 overview Um, so before I exit out of the slideshow, is there any questions for me about any topic? I can go back to the topic list. I know it was really, really fast. Again, I did record it um, in case you want to go back and rewatch something because you couldn't keep up with me. Um, I understand. Or I already got asked if I would post the Jeopardy. I can actually post the link to the Jeopardy itself and the questions. So I will post that as soon as this is over. Um, and if you are not in my class, because not all of you are, I probably will not be able to get in contact with your teachers before tomorrow morning fast enough to get them to post the Jeopardy. So if you message me on Schoology, um, I can send you the link as well. So it'll probably be under my name, Kara Zortman. Um, but yeah, just look for Zortman on Schoology and you can ask me for the link. Okay, I'm seeing questions. Oh, Daniel, you didn't see. Someone did say you got this, but it was a direct message to me, so I don't know if you saw it. You're
Have
not
you ever said that?
You guys got this. I will say just really take your time with the questions. Usually there are two in every question that are pretty far off from what they're asking about. And those are pretty easy to identify most of the time. And then the two that it usually comes down to are like there's one that will definitely obviously be very correct as long as you know it. The other one's usually like the opposite of what the answer is. Um, so I don't know what to expect fully. This is the first online assessment, so there's a little bit more leeway here for them to ask different questions, but just trust yourselves. That's all I can recommend. You all put in work this year. You're all here right now. Um, just go into it with as positive as an attitude as you can, and that's all you really can do at this point, okay? The better you make yourselves feel about it, the more you convince yourselves that you got it, the better you're likely to do. My dog thinks so too, and the guinea pigs.
Thank you, Miss Norman.
That scares me. Is there anything else anyone needs? At that rate, how are you writing on my screen? Who gave Daniel permission to write on my screen? What kind of crazy... Okay, I'm gonna stop sharing my screen now because it's being written on. What in the new Zoom feature was that? All right. So that's all I got. So you don't have to hang out in here any longer. If you have any questions you're waiting to ask me, you can feel free to stay on. I will sit here patiently until most of you hop off. But if not, have a good night. Again, message me if you need anything, and I will be rooting for you.
Thanks, Mrs. Ortman.
Of course.
Thanks, Ms. Zortman.
You're welcome. We got our crew here. You have a good night as well. Sleep good. You need it. All right. That's